In this session, we are going to discuss about the evaluation of antimicrobial agent effectiveness. That is the evaluation of the chemical antimicrobial agent, and this is the testing of disinfectants. Chemical antimicrobial agents or disinfectants used in hospitals and laboratories must be periodically tested to determine their efficacy. The disinfection process validation, it is defined as establishing a documented evidence that a disinfection process will consistently remove or inactivate known or possible pathogens from inanimate objects. This is termed as the disinfection process validation. And this testing of antimicrobial agents, it is regulated by two different agencies. For the regulation of the testing of disinfectants, it is regulated by the US Environmental Protection Agency, whereas agents used on humans and animals are controlled by the Food and Drug Administration. There are several methods for testing disinfectants. Each of these methods have their own advantages and disadvantages. And these tests are carrier test, suspension test, capacity test, practical test, in-use test or field test. So these are the different methods for testing the effectiveness of a disinfectant. Now we are going to discuss about the carrier test. These are the oldest test and it was described in 1881 by Robert Koch. In this particular test, a carrier will be used. The carrier may be either a silk or a catgut thread or a penny cylinder, that is a little stick. And this carrier will be contaminated by submersing into a test organism culture. A liquid culture of the test organism will be used to contaminate this particular carrier. When this test was originally described by Robert Koch in 1881, he used a silk thread and the contaminating organism used by him was Bacillus anthracis. So once the carrier is immersed into the liquid culture, it will be then dried, and after drying it will be brought into contact with the disinfectant to be tested, and it will be brought in contact for a particular duration of time, for a particular exposure time. After the exposure time, the carrier will be cultured in a nutrient medium. If there is no growth in the nutrient medium, that indicates that the disinfectant tested have high bactericidal activity, or if there was growth in the nutrient medium from the carrier, that indicates that the disinfectant was not that much effective. Example for a carrier test is a use dilution test. The carrier tests have a few limitations. They are to have a few disadvantages. They are the number of bacteria dried on the carrier cannot be standardized. We cannot standardize the number of bacteria. Also, the survival of bacteria during the drying of carrier is also cannot be kept constant. Next is the suspension test. In this test, a sample of the bacterial culture is directly suspended in the disinfectant solution. It will be kept so for a period of exposure time, and after the exposure, it will be analyzed to identify whether the inoculum or the organism is killed or not. The suspension test is preferred to carrier test, as here the bacteria are uniformly exposed to the disinfectant. Also, there is no problems associated with the survival of organisms during the drying of the carrier. There are different types of suspension test. They are the qualitative suspension test, the quantitative suspension test, and the phenol coefficient test. In a qualitative suspension test, a loop full of the bacterial suspension will be brought into contact with the disinfectant for a period of time, and after this period of exposure, the mixture will be cultured to observe for the surviving microorganisms. The results will be expressed as growth or no growth. Growth indicates that the disinfectant is not effective, and if there is no growth of microorganisms after their contact with the disinfectant, that means the disinfectant is highly effective. The quantitative suspension test is also performed, same as above, but here instead of just observing for the growth or the no growth pattern, the number of surviving organisms will be counted, and the surviving organism number will be compared to the original inoculum size to obtain a more quantitative assessment of the disinfectant efficiency. Next is the phenol coefficient test. Phenol coefficient is a term used to denote the efficiency of a particular disinfectant. It is calculated by dividing the dilution of the test disinfectant by the dilution of phenol that disinfect under predetermined conditions. That is, we will observe the different dilutions of test disinfectant which can kill the microorganisms, and this particular dilution will be divided by the dilution of phenol that also disinfects or kills the microorganisms under similar conditions. And this particular calculation will give us the phenol coefficient. The phenol coefficient can be determined by different methods, and they are Rideal Walker method, Chick-Martin test, Garrod test, etc. Now, Rideal Walker method. Here, the phenol and the test disinfectant are diluted to different concentrations, and their bactericidal activity is determined by using a microorganism, by using a bacteria, and the test organism generally used is Salmonella typhi. So the Salmonella typhi will be suspended in different concentrations of phenol and different concentrations of the test disinfectant, and they will be incubated for different time intervals. The time intervals generally adopted are 2.5 minutes, 5 minutes, 7.5 minutes and 10 minutes. After these time intervals, the microbial survival will be observed by inoculating them into a nutrient medium, and after incubation at 37 degrees Celsius for 48 to 72 hours, the microbial survival, the number of microorganisms growing in these plates will be observed, and we can identify the dilution of the test disinfectant which disinfects or kills the microbial suspension in a given time, and also we can also observe the dilution of phenol which can also kill the microbial suspension in the same time. So we will observe both the dilution of the disinfectant, test disinfectant, and the dilution of the phenol which are capable of killing the suspension under the same conditions. So by dividing the dilution of test disinfectant by the dilution of phenol, we can calculate the phenol coefficient. So if the effective dilution was 1:90 for phenol and in 1:450 for the disinfectant, the phenol coefficient can be calculated as 450 divided by 90, which is equal to 5. So the phenol coefficient for this test disinfectant is 5. This Rideal Walker method for determining the phenol coefficient has several disadvantages. They are: there is no organic material included in the test. In real life scenarios, the antimicrobial action of the disinfectant will be effective, should be effective in the presence of several organic materials. Such a test condition is not included in Rideal Walker method. It is a disadvantage. And we are using a single microorganism, Salmonella typhi only, and it may not be appropriate. And the time allowed for disinfection, we are using a maximum of 10 minutes incubation, and it is very short. And this Rideal Walker method can be used to evaluate phenolic type disinfectants only. Now we will discuss the second type of phenol coefficient test, that is Chick-Martin test. This test also determines the phenol coefficient of the test disinfectant as that of the Rideal Walker method. But here, to simulate the presence of organic matter, a 3% of dried human fecal matter will be added along the test. So there is addition of organic material here, and the effectiveness of the disinfectant will be tested in the presence of this particular organic matter. And also the exposure time is fixed at 30 minutes. And the test organisms used are Salmonella typhi and Staphylococcus aureus. Both gram-positive and gram-negative organisms are used as a test organism here. Next method of phenol coefficient test is Garrot's test. This is also a similar phenol coefficient test. This is a modified Chick-Martin test. Here, the organic material is added, which is a 5% of yeast suspension instead of dried human fecal matter. In Garrot's test, 5% of yeast suspension is added as an organic material. Now, we will discuss about the capacity test. The capacity of a disinfectant is known as the ability to retain the antimicrobial activity even in the presence of an increasing load of dirt and bacteria. So, to test for the capacity of an antimicrobial agent, in a capacity test, the disinfectant is challenged repeatedly by successive additions of bacterial suspension. Kelsey-Sykes test is a capacity test. Kelsey-Sykes test is a triple challenge test. That means there will be 3 successive additions of microbial suspension into the disinfectant. And this test can be done under both clean and dirty conditions. The test is done in hard water. The hard water will be used for preparing the disinfectant dilutions, and this hard water is used for testing under clean conditions, and yeast suspension is used for testing under dirty conditions. The test organisms that are used for this test include Staphylococcus aureus, Pseudomonas aeruginosa, Proteus vulgaris, or E. coli. As we said before, there will be 3 successive additions of the test bacteria. They will be added at the 0th minute, 10th minute, and 20th minute. And after each addition of this test bacteria, it will be kept for 8 minutes incubation, and after this 8 minute, the survival of the bacteria will be observed by inoculating into a nutrient medium. And the disinfectant will be declared to be an effective disinfectant if there is no bacterial growth observed after the first and second challenge of bacterial addition. If the disinfectant is found to be effective after capacity test, then its stability upon storage should be determined. And this is done by the stability test or the supplementary test. So if the prepared disinfectant solutions are to be kept for more than 24 hours, its stability should be measured. For this test, the test organism used will be Pseudomonas aeruginosa. And for performing this test, the disinfectant solution will be prepared in 2 sets. The first set will be inoculated with the test organism, that is Pseudomonas aeruginosa, and it will be incubated for 7 days. And after this 7-day incubation, it will be tested for bacterial growth by inoculating into nutrient media, and it will be observed. In the second set of the disinfectant solution will be kept aside at room temperature for about 7 days, and after the 7 days of keeping aside, it will be inoculated with the test organism, and it will be tested for the survival of the bacteria by inoculating into a nutrient medium. If the bacteria survive the particular dilution of the disinfectant tested in the stability test, then that means the particular dilution of the disinfectant is not stable for storage. So a higher concentration must be tested by performing this particular supplementary test or stability test. Next is the practical test. We are determining the effectiveness of the antimicrobial agent in laboratory level experiments. The laboratory level effectiveness may not always correlate with that of the real life situations. So the practical tests are done to evaluate the effectiveness of the disinfectant under real life conditions. It is performed to measure the time concentration relationship of the disinfectant. It is done by performing a quantitative suspension test. So the objective is to identify whether the dilution or the concentration of the disinfectant that we are going to use is adequate in the real life conditions under which it is going to be used. We can use the surface disinfection test, which assess the effectiveness of the selected agent against surface adhere microorganisms. So we will use different types of test surfaces, which may be a small tile, a microscopic slide, a piece of PVC, a stainless steel disc, etc., which will be contaminated with the test bacteria, and then it will be dried. Then a definite volume of the disinfectant solution will be distributed over this particular carrier, and after a particular period of exposure time, the number of survivors on the carrier will be determined. If there are more number of microorganisms surviving, then that means the disinfectant is not that much effective against the test bacteria. So this test is different from that of the carrier test. In the carrier test, the carrier is submerged in the disinfectant solution during the whole exposure time, and the carrier will be having the microorganisms dried onto it. Whereas in the surface disinfectant test, the disinfectant is applied onto the carrier. The carrier is not immersed in the disinfectant here. Instead, on the surface of the carrier, the disinfectant is just applied. It is not immersed in the disinfectant during the entire exposure time. So these are the differences between the carrier test and the surface disinfection test. The surface test can reflect the in-use, real use conditions like contact times, temperature, use dilutions, surface properties, etc. Next is surface time kill test. Here, a 24-hour culture in a nutrient medium is prepared and spread onto the surface of a sterile test surface, and then this inoculated test surface are treated with the disinfectant for different durations of time. And after the treatment time, the test surfaces are placed into a neutralizing solution which will neutralize the disinfectant, and then the number of surviving microorganisms are determined by culturing. Here, an untreated inoculated test surface is kept as the control. Next is the in-use test. This was first described in 1985 by Maurer. This is used to detect the contamination of disinfectants. Here, a 1 ml sample of the disinfectant will be added to a 9 ml of diluent. This diluent will contain a particular inactivator. And after this particular mixing, 10 drops of this diluted disinfectant will be placed on 2 nutrient agar plates. One plate will be incubated at 37 degrees Celsius for 3 days. The other will be incubated at room temperature for 7 days. And if you observe, if 5 or more colonies on these plates, that means that the disinfectant is contaminated. It cannot be further used. So, we will discuss the testing scheme for disinfectants. The antimicrobial efficiency of a disinfectant should be examined at least at 3 stages of testing. First phase involves laboratory test, during which the chemical agent is having any antimicrobial activity. This is generally done by performing quantitative suspension test. During the second stage, which is generally carried out in the laboratory using conditions that simulate real life conditions, here generally practical tests are more of the common performed. In the third phase of disinfectant testing scheme, field tests and in-use tests are performed to further confirm the antimicrobial activity of the disinfectant. And the antimicrobial activity of a disinfectant is assessed in terms of activity towards vegetative bacteria. If it is having activity against vegetative bacteria, it is to be determined by bactericidal test. Activity towards fungi and yeast are determined by performing fungicidal test. Activity against mycobacteria are determined by performing tuberculosidal test. And virucidal tests are performed to to evaluate the antiviral effectiveness of the disinfectant. And sporicidal tests are performed to find out whether the disinfectant is having activity against spores of bacteria. So, in this session, we discussed about various techniques for evaluating the antimicrobial agent effectiveness. We discussed carrier test, suspension test, capacity test, practical test, in-use test, etc. Thank you so much for listening.